Section 3 of Gray's Anatomy, Part 4. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by M.L. Cohen. Anatomy of the Human Body, Part 4 by Henry Gray. Development of the Nervous System, Part 1. Development of the Nervous System. The entire nervous system is of ectodermal origin, and its first rudiment is seen in the neural groove, which extends along the dorsal aspect of the embryo. By the elevation and ultimate fusion of the neural folds, the groove is converted into the neural tube. The anterior end of the neural tube becomes expanded to form the three primary brain vesicles. The cavity of the tube is subsequently modified to form the ventricular cavities of the brain and the central canal of the medulla spinalis. From the wall of the nervous system, elements and the neuroglia of the brain and medulla spinalis are developed. The medulla spinalis. At first, the wall of the neural tube is composed of a single layer of columnar ectodermal cells. Soon the side walls become thickened, while the dorsal and ventral parts remain thin and are named the roof and floor plates. A transverse section of the tube at this stage presents an oval outline, while its lumen has the appearance of a slit. The cells which constitute the wall of the tube proliferate rapidly, lose their cell boundaries, and form a syncytium. This syncytium consisted first of dense protoplasm with closely packed nuclei, but later it opens out and forms a looser meshwork, with the cellular strands arranged in a radiating manner from the central canal. Three layers may now be defined: an internal or ependymal, an intermediate or mantle, and an external or marginal. The ependymal layer is ultimately converted into the ependyma of the central canal. The processes of its cells pass outward towards the periphery of the medulla spinalis. The marginal layer is devoid of nuclei and later forms a supporting framework for the white funiculi of the medulla spinalis. The mantle layer represents the whole of the future gray columns in the medulla spinalis. In it, the cells are differentiated into two sets, that is, spongioblasts, or young neuroglial cells, and germinal cells, which are the parents of the neuroblasts, or young nerve cells. The spongioblasts are at first connected to one another by filaments of the syncytium. In these, fibrils are developed so that as the neuroglial cells become defined, they exhibit their characteristic mature appearance with multiple processes extending from each cell. The germinal cells are large, round or oval, and first make their appearance between the ependymal cells on the sides of the central canal. They increase rapidly in number, so that by the fourth week they form an almost continuous layer on each side of the tube. No germinal cells are found in the roof or floor plates. The roof plate retains, in certain regions of the brain, its epithelial character. Elsewhere, its cells become spongioblasts. By subdivision, the germinal cells give rise to the neuroblast or young nerve cells, which migrate outward from the sides of the central canal into the mantle layer and neural crest, and at the same time become pear-shaped. The tapering part of the cell undergoes still further elongation and forms the axis cylinder of the cell. The lateral walls of the medulla spinalis continue to increase in thickness, and the canal widens out near its dorsal extremity and assumes a somewhat lozenge-shaped appearance. The widest part of the canal serves to subdivide the lateral wall of the neural tube into a dorsal or alar and a ventral or basal lamina, a subdivision which extends forward into the brain. At a later stage, the ventral part of the canal widens out, while the dorsal part is first reduced to a mere slit and then becomes obliterated by the approximation and fusion of its walls. The ventral part of the canal persists and forms the central canal of the adult medulla spinalis. The caudal end of the canal exhibits a conical expansion, which is known as the terminal ventricle. The ventral part of the mantle layer becomes thickened, and on cross-section appears as a triangular patch between the marginal and ependymal layers. This thickening is the rudiment of the anterior column of gray substance and contains many neuroblasts, the axis cylinders of which pass out through the marginal layer and form the anterior roots of the spinal nerves. The thickening of the mantle layer gradually extends in a dorsal direction and forms the posterior column of gray substance. The axons of many of the neuroblasts in the alar lamina run forward and cross in the floor plate to the opposite side of the medulla spinalis. These form the rudiment of the anterior white commissure. About the end of the 4th week, nerve fibers begin to appear in the marginal layer. The first to develop are the short intersegmental fibers from the neuroblast in the mantle zone, and the fibers of the dorsal nerve roots, which grow into the medulla spinalis from the cells of the spinal ganglia. By the 6th week, these dorsal root fibers form a well-defined oval bundle in the peripheral part of the alar lamina. This bundle gradually increases in size, and spreading towards the middle line, form the rudiment of the posterior funiculus. The long intersegmental fibers begin to appear about the 3rd month, and the cerebrospinal fibers about the 5th month. All nerve fibers are at first destitute of medullary sheaths. Different groups of fibers receive their sheaths at different times. The dorsal and ventral nerve roots about the 5th month, and the cerebrospinal fibers after the 9th month. By the growth of the anterior columns of gray substance, and by the increase in size of the anterior funiculi, a furrow is formed between the lateral halves of the cord anteriorly. This gradually deepens to form the anterior median fissure. The mode of formation of the posterior septum is somewhat uncertain. Many believe that it is produced by the growing together of the walls of the posterior part of the central canal, and by the development from its ependymal cells of a septum of fibrillated tissue which separates the future funicula gracilis. Up to the 3rd month of fetal life, the medulla spinalis occupies the entire length of the vertebral canal, and the spinal nerves pass outward at right angles to the medulla spinalis. From this time onward, the vertebral column grows more rapidly than the medulla spinalis, and the latter, being fixed above through its continuity with the brain, gradually assumes a higher position within the canal. By the 6th month, its lower end reaches only as far as the upper end of the sacrum. At birth, it is on a level with the 3rd lumbar vertebra, and in the adult with the lower border of the 1st or upper border of the 2nd lumbar vertebra. A delicate filament, the filum terminale, extends from its lower end as far as the coccyx. The spinal nerves. Each spinal nerve is attached to the medulla spinalis by an anterior or ventral and a posterior or dorsal root. The fibers of the anterior roots are formed by the axons of the neuroblast which lie in the ventral part of the mantle layer. These axons grow out through the overlying marginal layer and become grouped to form the anterior nerve root. The fibers of the posterior roots are developed from the cells of the spinal ganglia. Before the neural groove is closed to form the neural tube, a ridge of ectodermal cells, the ganglion ridge or neural crest, appears along the prominent margin of each neural fold. When the folds meet in the middle line, the two ganglion ridges fuse and form a wedge-shaped area along the line of closure of the tube. The cells of this area proliferate rapidly opposite the primitive segments and then migrate in a lateral and ventral direction to the sides of the neural tube, where they ultimately form a series of oval-shaped masses, the future spinal ganglia. These ganglia are arranged symmetrically on the two sides of the neural tube and, except in the region of the tail, are equal in number to the primitive segments. The cells of the ganglia, like the cells of the mantle layer, are of two kinds, that is, spongioblasts and neuroblasts. The spongioblasts develop into the neuroglial cells of the ganglia. The neuroblasts are at first round or oval in shape, but soon assume the form of spindles, the extremities of which gradually elongate into central and peripheral processes. The central processes grow medialward, and becoming connected with the neural tube, constitute the fibers of the posterior nerve roots, while the peripheral processes grow lateralwards to mingle with the fibers of the anterior root in the spinal nerve. As development proceeds, the original bipolar form of the cells changes. The two processes become approximated until they ultimately arise from a single stem in a T-shaped manner. Only in the ganglia of the acoustic nerve is the bipolar form retained. More recent observers hold, however, that the T form is derived from the branching of a single process which grows out from the cell. The anterior